So first I want to thank everyone for coming and for the panel for obviously selecting me, so this is pretty cool. So again, I'm just going to discuss some of the work that I did in Mustafa's lab while I was a grad student with him. In particular, how we developed using nuclear targeted gold nanoparticles to monitor different biological processes in real time. But before we get into the science, we were asked to give a little bit of a background about ourselves. And I think this slide pretty much sums it up. I'm a person of two distinct passions. I really love running, and I really love science. So while I was completing my undergrad degree at the University of Central Florida, I was also competing on the national and international level in several track and field meets. In particular, I was able to represent the US in both 2003 and 2007 at some pretty large international competitions, and I was actually ranked 5th in the country in 2007, which then qualified me for the 2008 Olympic trials. Unfortunately, I didn't make the 2008 Olympic team, so my next plan was to take a break. And what better way to take a break than to decide to go to grad school? So that's what I chose to do, and it's seemingly working out well for me. But I also wanted to, before I get into all the science that I did, really acknowledge the people that helped to get me to where I am today. So like I said, I did my undergraduate degree at the University of Central Florida under the direction of Dr. Trine Ho. She was so instrumental in making me the scientist I was today and really introduced me to gold nanoparticle research. From there, I did my PhD work with Dr. Mustafa El-Sayed, and I had a great 5 years, and he really pushed me to be the best scientist I could be. While there, I was also fortunate enough to collaborate with a postdoc, Ben Kang, and he's co-author on a lot of the work that I'll show today. From there, I decided to do a postdoc up at Harvard Med School and Mass General Hospital. And I think one of the, well, there's two major things. One, I got to see a surgery, which is really cool for me, because as a, a basic research scientist, sometimes we don't get to see that. So that was really cool. But one of the other major impacts of that postdoc was we had an awesome collaboration with the two individuals on the right, Peter and Petra. They dubbed themselves as serial entrepreneurs. And that was for really the first time that I started to consider a career in industry. Petra was really successful in industry, and I really saw what a strong female scientific figure could do in an industry setting. And so that then took me to Merck, where I'm currently a senior scientist. But to get into the science, throughout my career, I've always utilized gold nanoparticles. So I just wanted to give a little bit of a background of why I enjoy working with gold nanoparticles and why I think they're so great. If we look at this scale here, we can see that a lot of important biological molecules lie in the range between 1 and 100 nanometers. And it's no coincidence that these gold nanoparticles, being dubbed nano, also lie in this range. And so they're comparable in size to important biological molecules, and therefore can interact with these molecules. But in particular for gold nanoparticles, being a metallic nanoparticle on this nano scale, they have unique optical and physical properties. And in particular, they're able to strongly scatter light, much, much more efficiently than typical fluorophores or conventional fluorophores. So we can actually leverage that characteristic when we're monitoring and detecting low levels of biological molecules in potentially complex environments. So when we wanted to set out and develop a system that we could monitor biological processes in real time, we needed to create a probe that could allow us to do that. And so for that, we synthesized gold nanoparticles and functionalized them with peptides that would allow them to be internalized into the cell, but also localized at the nucleus where a lot of these important events are occurring. And to leverage the strong light scattering, that allowed us to, one, collect Rayleigh scattering from these cell samples. And so in that way, we're able to acquire morphological information of these cells over time using just a conventional dark field microscope. But additionally, because these particles scatter light so strongly, they're able to amplify rather low signals, such as Raman vibrations. So here we can actually get a molecular fingerprint of these cellular events by monitoring the Raman spectra over time. So we were able to apply this nuclear-targeted gold nanoparticle Raman setup to a bunch of different intercellular processes. But I really wanted to focus on these two applications that we looked into, evaluating drug efficacy over time, as well as monitoring drug delivery in real time. So in terms of evaluating drug efficacy, we first needed to see, one, can these nanoparticles localize at the nucleus, which we can see in this inlay here, is possible, but also get an amplified Raman signal. And that's what we see here in this Raman spectra. In the biological fingerprint region, we get very clear, distinct signals. And if we take these nuclear-targeted treated cells and treat them with common chemotherapeutics such as cisplatin or 5-fluorouracil, we can see distinct changes over time. In particular, if we look at the cellular imaging, we can see that our cells have now started to shrink and the light scattering has been amplified, and this is indicative of a cell undergoing cell death or apoptosis. But more importantly for us, since we really wanted to monitor the molecular changes over time, if we look at these spectra, we can see that there are distinct vibrational changes that occur after therapeutic treatment. In particular, we have a decrease in the 500 wave number band, but we also get a simultaneous increase in the 1,000 and around 1,600 wave number region. And so we were actually able to correlate these changes to distinct alterations in these biological molecules. So for the first two vibrations at 500 and 1,000, that's actually representing proteins denaturing while these cells are undergoing cell death. And the higher wave number band at 1585 is actually indicating that there's possible DNA damage occurring during cell death. And I didn't show it here, but we were able to actually compare this process to typical cell viability assays such as XCT and MTT. And because we have these nuclear targeted particles which are able to amplify these signals on the molecular level, we're actually able to get out drug efficacy comparisons between different chemotherapeutic drugs in a shorter time frame. So as a grad student who wanted to graduate, I found this pretty appealing. But another thing that we were able to do was to show that we were able to track drug delivery over time. So we took our nuclear-targeted gold nanoparticles, but also functionalized them with a common chemotherapeutic drug, doxorubicin. It has a distinct Raman vibration, but also is fluorescent on its own. And so when doxorubicin is functionalized to the surface of these gold nanoparticles, the Raman vibration of doxorubicin will be greatly enhanced because of the strong light scattering of these gold nanoparticles. But also, because these nanoparticles have unique optical properties, the fluorescence is actually quenched of this doxorubicin. And so the linker that we chose to use was pH sensitive, so that when these cells when these gold nanoparticles are actually internalized into the cell, and they localize into the lysosome before heading to the nucleus, this pH sensitive linker will be cleaved, therefore releasing the doxorubicin molecule, and therefore the doxorubicin fluorescence will be restored. However, the Raman vibrations will actually be decreased. And so we could see that we were able to track this over time, where when we initially treated the cells, we can see a strong Raman vibration of doxorubicin. However, its fluorescence is quenched. But as these nanoparticles travel through the cell into different organelles, specifically the lysosome where the pH drops, we see that this Raman signature disappears. However, the fluorescence is restored. So with this, we were able to monitor drug delivery in real time, which we thought was pretty exciting. So in terms of what's next, since I'm in the pharmaceutical world now, being able to miniaturize and automate the system would be really advantageous. The system that we were using was a bulk regular microscope that was modified to have dark field optics and have a live cell chamber. But if we could imagine doing this in a 96 well plate, we could do high throughput drug screening. And since now I'm working in the discovery space, we're triaging a bunch of molecules in a short time period. So this could be really, in terms of interest, at least for me personally. In terms of in vivo applications, if we could employ our drug delivery and localization application to an in vivo setting where we're trying to assess tumor targeting as well as seeing whether a certain chemotherapeutic is actually efficacious in that environment, that would be a really great step forward. But also there have been some groups that are not only using it to identify specific tumors, but they're also using it as an interoperative tool where they're using high Raman signals within the tumor to actually resect the tumor margins to make sure that they're actually eliminating and removing the tumor and the diseased tissue, but not removing too much. So with that, I'd like to thank the people that helped me achieve this great honor, Mustafa El Sayed in particular, as well as CNE News and ACS for this awesome opportunity.